Hi, everybody. We're here for what is the second the John Martin Memorial Lectureship. Thanks for coming. I'll start out. We have a really terrific program today, both with folks who are going to make comments about John at the beginning and end of the lectures, and two really outstanding scientific lectures. Bill will kick it off. Bill Lee was an executive vice president of research at Gilead during what I will say were Gilead's golden years, which Bill contributed to enormously. Really outstanding scientist with a huge record of accomplishment in bringing antiviral therapies to the world. He's also a pool hustler, okay, but we could talk about that later, okay? But a terrific person and a real friend of Scripps. Bill, I'll turn it over to you.
All right. We definitely need to change that slide. And Pete, this is actually the third memorial. So, good afternoon. That's my first line, actually. Good afternoon, everyone. And my name is Bill Lee, and welcome to the third edition of the John C. Martin Memorial Lectureship at Scripps. Now, this lecture was established through the generosity of John's friends, numerous friends, colleagues, both at Scripps here and around the globe. And your support, if you're one of those people in the audience of this lectureship, is profoundly appreciated by all, and I thank each one of you, those present today and those who are unable to join us. So our focus today, as in previous years, revolves around the exploration of fundamental biology and its application to the discovery of new medicines, and to treat patient unmet medical needs. And this is really what John Martin was about. Now, I'll come back to that. In the first edition of this lecture series, Dr. Rich Whitley, spoiler, he's the closer today, Norbert Bischofberger and Dr. Peter Schultz hammered on these themes: revolutionary biology and innovative drug discovery. The following year, lectures by Thomas Solar and David Liu provided groundbreaking insight into HIV biology, innovative drug discovery, and advancements in vivo genome editing. Today's lectures promise to uphold this high standard from Dr. Owen Witte, professor of microbiology, immunology, and molecular medicine. And Owen, I think there's a number of other departments you're involved with, but I'll let Hugh talk about those. And Dr. Xiaowei Zhuang, professor of chemistry and chemical biology at Harvard, and Dr. Arden Pataposhian will introduce her. So at last year's lectureship, I gave the closing remarks, and I shared some memories, photos, and a few stories about John, perhaps with a bit too much levity. Because I opened the session today, I'm going to try to be a little bit more respectful, less fun, but I still have some stories. And so if I could have the first slide. Okay. This is actually one of my, maybe I'm going back on that, making fun of John, but this is actually one of my favorite pictures of John. Thomas Solar, who's in the audience today, John and I had just flown 14 hours from San Francisco to Prague, where John was scheduled to give a lecture and receive an award the following morning at the Institute of Organic Chemistry and Biochemistry. It was here at this institute in the mid-'80s, with the backdrop of the Iron Curtain, that John began a long and fruitful collaboration with Anton Holy. At the institute, it was Holy and his group that first synthesized the acyclic nucleotide analogs, which later became the basis of 4 Gilead drugs and became the backbone of another 10 drugs. So instead of heading directly to our hotel and relaxing after a long flight, like any normal person would do, John insisted that we go to a beer cellar in the city center. In this picture, John he's on his phone prepping for his upcoming lecture and asking us various questions. John had recently started taking all of his notes on his iPhone. And this was an evolution from the first 20 years at Gilead, when he famously managed everything, his thoughts, questions, reminders, on a little black notebook. I'll come back to this. So in the world of pharma and biotech, John was one of those vanishing breeds of scientist CEO. He was truly visionary, leading Gilead from a concept to a global leader in the treatment of HIV, HCV, and HBV. But he was more than just a successful executive. He was a scientist at heart whose decisions began and ended with patient welfare. What made John extraordinary, beyond his business acumen, was his early initiation of a global drug access program. He believed that all patients should have access to life-saving drugs, regardless of their ability to pay. He did this before Gilead was profitable. Profitable. And remember, this was at a time when Big Pharma was fighting patent infringements in South Africa, when the company was being ravaged by HIV. That global access program, which was led by Clifford Samuels, who's also in the audience today, has treated over 40 million patients in the developing world for HIV, HCV, and hepatitis B. This program distinguishes him not just as a great CEO, but as a humanitarian. And it's for these reasons that John, I strongly believe, is the GOAT of biotech CEOs. GOAT being the greatest of all time. So. All right, that's enough nice things about John. All right. John's favorite way to experience new science was on the floor. At every conference, John was the first person in the door when the posters were put up. He always wanted to hear science from the people doing the science. He wanted to experience their passion, their doubts, and, of course, the details. Always taking notes, always writing questions in his little black notebook. Posters were required reading at Gilead. And it was at a poster at an EASL meeting, a European Liver meeting, in 2011 that convinced John that we needed the asset sofosbuvir, which later became the drug Sovaldi for the treatment of HCV. Pharmasset had presented early data, early phase 2A data, and we had been working along with many other pharma in this space, and he realized, and we realized, that this was an asset we had to have. And 6 months later, for $11 billion, the largest biotech acquisition at the time, we bought the company, Pharmasset. Now, and remember, this was a very competitive space, the HCV space, but the reason we got it is because John and the senior R&D staff studied that poster. And I doubt at that time there were any other CEOs at the poster, or at least none of them spent as much time as John spent at that poster. So, all right, one story. That was a story, but now another story. So at ASLD several years later, this is in Boston, this is the American Liver Disease meeting, 2013, John Link, Vice President of Medicinal Chemistry, and Bill Delaney, Senior Director of Biology at Gilead, were returning to the Back Bay Hilton, and that's where we always stayed. And they had spent a whole day with corporate development, going to various related meetings outside of scientific conference, and those meetings were always a big distraction on your conference schedule. So they walk into the bar, and I know it sounds like a joke, and at the bar, there's John seated. Okay, and in their minds, what better time to mingle with the CEO after a full day of doing corporate development work? Well, they sit next to John, and within a few minutes, John pulls out his little black book, and he begins a 45-minute interrogation of all the posters that he has seen and they have seen none of. We had a term for this at Gilead, and it was called Martinizing. and they got it, both barrels. But so the next morning, I see John Link and Bill. They were still in shock, and their question to me was they just wanted to know if they still had a job, because they were Martinized, and they still did have a job. So their take-home lesson, all of our take-home lesson at Gilead R&D was always go to the poster. And if you saw John take out his little black book, head for the door, any door. All right, well, that is my opening remarks, trying to be a little serious. Thank you for your attention. Without further ado, please join me in welcoming Professor Hugh Rosen to the stage, who will introduce our first speaker, Owen Witte. Thank you.
Thanks, Bill, for the introduction. It's a very special privilege for me to introduce Owen Witte as the first keynote speaker in today's John Martin Memorial Lectures. Owen and his group have made a number of seminal contributions to the understanding of leukemias, immune disorders, and most recently, epithelial cancers. Following a Stanford MD, Owen's scientific trajectory really went stratospheric during a postdoc with David Baltimore, when he described tyrosine kinase activity for ABL, and then illuminated the role of BCR-ABL oncoproteins in human leukemias. And this mechanistic work opened up the possibility of treating chronic myeloid leukemias with Gleevec. He subsequently discovered BTK and its role in X-linked agamaglobulinemia, and these two examples really have been the leading edges of a monumental body of work. He has been a long-standing faculty member at UCLA. He's a member of the National Academy of Sciences and the National Academy of Medicine, and has really had more awards and prizes than I could possibly mention. As a physician and a scientist, he has really brought unsurpassed integrity and imagination to impact on both basic science and therapeutics. And this, I think, makes him the perfect lecturer to honor the life and the legacy of John Martin. Owen Witte.
Good afternoon. It is a pleasure to be here, and thank you very much for the invitation. I didn't know John Martin as well as some of you in the audience, but I got to know him, and I'll tell you that story as we get into the slides. But I thank you for the introduction, and he's accurate when he said that I've had a lot of interaction with the biotechnology and pharmaceutical industry, including this laundry list of companies that I've either consulted for, helped found, and so on. And there's a reason I put this on the first slide. Sometimes people will put their affiliations, non-academic affiliate, on a second slide and then zip through it in 2 microseconds, as if they are embarrassed about it. I'm not only not embarrassed about it, I think this is possibly the best training environment I've had in the last 35, 40 years, which is to think about our science in light of what might be a practical application to the treatment of human disease. Let's see if I can figure out how to, here we go. There, okay. This is a picture taken from the early days of a company called Kronos Bio. You can see John in the back of the table there. No, it's apparently not, okay. There's 2 slides in front of me here. This one is, all right. Here's John, myself, Ari Beldegren, others. And this was in the early days of Kronos, and some of you, I'm sure, know Norbert Bischofberger, who was also previously associated with Gilead. This wasn't the first time I met John. The first time I met him was when Gilead was investigating purchasing Kite Pharma, of which I was proud to be a founder and in charge of the scientific advisory board. John came with a few key members of his staff to visit what was then the recently purchased main research center for Kite in Santa Monica, purchased from a company, a Japanese pharma that had bought a previous company called Agensis that was also a founder of. And John was there to look, to learn, to ask questions. And he took me almost aside and didn't ask about the building, didn't ask anything about the corporate structure of Kite and so on. He really wanted to know about the science and what I thought the future of this cell-mediated therapy of cancer was all about. And it was the most interesting and engaging discussion while we walked through the corridors and back and forth and this talk. And I realized that I had just been so beautifully interrogated, okay, and he had sucked out just every important piece of information, okay, that I possibly had in my head about Kite, what our plans were, what our products were going to be, et cetera. And then afterwards, I was really glad that Gilead consummated the deal because I had basically given away all the trade secrets of what was next. I can't remember. The amount of money was just enormous. It was a wonderful payday for everybody associated with Kite. But more importantly, the products were taken forward and expanded, and there are thousands and thousands of patients that have benefited from the hand that John and Gilead played in taking Kite from a startup biotech into the pharmaceutical world and really delivered these medicines to many, many patients. And honestly, I'm as proud of that as I am of the other achievements of the company. All right. So I'm going to tell you a story, a combined set of stories about thinking of new ways to treat advanced epithelial cancers. But the perspective that I have on this goes back about 50 years to when I was a beginning medical student working in Irv Weissman's lab at Stanford on this particular problem, which is a specific retrovirus called the Maloney-Murine leukemia sarcoma virus complex that could induce sarcomas in susceptible strains of mice or rats with a very rapid kinetics, very aggressive pathology, but under the right circumstances could be regressed by action of the immune system to turn on myriad numbers of cytotoxic T cells that recognize the specific antigenic epitope generated by the virus and reduce those large tumors to nothing and provide lifelong protection to re-challenge with the same virus. Okay. So back then, we didn't know anything in detail about the genome structure. That was to come over a few years from then. But it was this pattern of aggressive pathology and activation of the immune system and regression that really captivated me. And I set out to say, if I could understand what caused this type of genetic change, okay, that would lead to this, and then understand what activated the immune system, that would be a good paradigm. And so everything that I've done subsequent to this in my career has really been thinking about these two sides of the equation: what drives the pathology, what causes the reaction of the immune system. And I won't talk about any of the nice work that you talked about that I've done over the years, but really talk about things that are much more current because that's what science should be, talking about the things you haven't yet finished rather than things that are long in the past. This is what I hope to be able to tell you today. I want to tell you about how we're modeling these very, very aggressive forms of prostate, lung, and other epithelial cancer types in our laboratory models. And this is a collaborative work with my former fellow, Jin-Wook Park, who's now at Duke, Tom Graber at UCLA. I'm going to tell you a little bit about how we're trying to develop T-cell receptor therapy to target a specific antigen that's very prevalent in early stage and mid stages of prostate cancer, prostatic acid phosphatase. Again, highly collaborative with John Lee at UCLA, Strong Lab, Wischlegel Lab, Heath Lab, Garcia Lab. And this is a macro trend in all of science, not just my own work, in which you really work collaboratively because of all the unique technical skills needed to get something done. And I'll bring up an area that I think is actually very cool called catch bonds, which is a way of making T-cell receptors highly effective for cytotoxic killing. And then finally, I'll tell you about what I think is a long-range project that I hope will go on far beyond when I'm retired from science, which is a way of using the change in information caused by processing of RNA differentially in cancer from normal cells to generate new antigenic epitopes that one can hopefully attack either with CAR T cells or, in the case I'll tell you about, with TCRs. So all of these projects relate to the basic biology of prostate cancer. Major epithelial cancer, hundreds of thousands of men at risk and under treatment, and it has an amazingly stereotypical biology despite having a very complex and combinatorial genetic predisposition and activation of mutations in the course of disease. Almost every man diagnosed with prostate cancer will have histologically adenocarcinoma of the prostate, 95% plus, if not greater. A very small percentage will be primarily diagnosed with a variant, sometimes usually referred to as neuroendocrine prostate cancer. Super aggressive, different than adenocarcinoma, and highly lethal. For the 95+% of men, if they're fortunate enough to have disease caught early enough, it can be treated with local therapy, surgery, radiotherapy, and that is often curative. But then there's a subfraction, and that can depend somewhat on Gleason grade, but not particularly on which specific genetic mutations have been documented in the primary lesions. And I think that's an interesting point. We'll come back with disease after some period of time, and that period of time can be very variable. It can be a few months and a return of the active disease measured by prostatic-specific antigen, or it can be literally years down the road, okay? And when that occurs, the application of a variety of chemical means, chemical castration or physical castration in some cases, to reduce the ambient signaling through the androgen receptor pathways, which are sustenance for prostate cancer cells at these early and mid-stage of the disease. Eventually, many men will progress through those therapies, and those therapies are non-trivial. They have very negative side effects, but they are valuable for keeping the tumor in check. But eventually, the disease runs into a phase called castration-resistant adenocarcinoma, and at this point, the tumor grows independent of any attempt to block androgen receptor activity or bypass androgen receptor activity, and eventually chemotherapy is tried. There have been some recent advances using PARP inhibitors and other things in the DNA repair pathway area, but they're certainly not curative, mainly suppressive, and usually death follows shortly after. Been studying prostate cancer for 25 years or so in different ways, worked with whole bunch of groups to develop animal models and also in vitro to in vivo models using tissue culture technologies and lentiviruses to introduce different genetic defects, and was happily studying the cellular components and the genetic rules for all of this when a different kind of experiment popped up in front of us. And this was an experiment in science administration, shown in this little block in the upper left corner. Two different groups, the Prostate Cancer Foundation and the Stand Up to Cancer group, had come together to decide to fund large team science to investigate particularly intractable problems in prostate cancer. And the most intractable problem was, what is the nature of castration-resistant prostate cancer and what might we do about it? So a group of multiple universities got together. In this case, it was called the West Coast Dream Team. We didn't give it that name, but that's the name. And it started in Vancouver, down to Oregon, UCSF, UCLA, Santa Cruz, and Davis. Investigators from all of these institutions. I was the co-PI on this particular program, project-style grant, with Eric Small from UCSF. We competed, we got the grant, and just one little aside, many other teams competed, but the other top team that competed against us was headed by Charles Sawyers, who's my former fellow, and Arul Chennaiyan, a colleague and friend, and we went to the finals for the competition for this fairly large grant, actually in Philadelphia under the auspices of the AACR. Chair of the committee to review these grants is Phil Sharp. And if you know Phil Sharp, he's just straight down what's going to be good, what's going to be bad. We went, we presented. Eric and I went back to the airport, and I got a phone call from the director of the Prostate Cancer Foundation telling us we had not gotten the grant. Okay? So this was a great thing to get on the plane with, okay? And heading home to Los Angeles, I get back to Los Angeles, and I have another phone call, and it's the same person calling, said, "We changed our minds, you're gonna get the grant." So this has never happened to me before or since. And what they decided to do is that they liked both teams so much, they got some, I don't know who, private donor to come forth with extra money for this grant. I think it was about $5 million grant. So that's probably the only thing you're going to remember from this seminar, okay, is that story. So we got into this, and the focus of this activity was castration-resistant prostate cancer is not normally a disease state in which you would get a biopsy. You already know the patient has cancer. You followed its course. You've got a great blood marker. Why do you need well, you need biopsies to learn something. So we worked out a very nice set of techniques with the interventional radiologist to get biopsies, and then we're going to do every omic you can imagine on them to try to learn something about what changed and what we might attack. Well, the very first set of biopsies really provided a dramatic set of clues as to what was happening. And this is simply at the histological level. And all of this is important to pathologists as they compare things, but just look from left to right, and you can see that there are different histologies associated under this rubric of castration-resistant prostate cancer, including in some biopsies regions that had this small cell carcinoma with neuroendocrine characteristics. Okay, and that's primarily at the H&E level, but with other confirming markers. But what was most interesting is that a single needle biopsy from a bone lesion, a liver lesion, anywhere, could have all three of these histologies in the same biopsy. And that suggested to me, as a biologist, we're looking at a differentiation schema, okay, which is happening stochastically around the body as different biopsies were taken from multiple patients. And the idea was that we're looking at a graded continuum, and that we have to understand what's driving that, and we have to understand what the stop points are or the sensitive points might be for developing new therapies. This idea and work from a bunch of labs became a very popular research area. And so the work I'll tell you about in the subsequent slides takes into account the fact that a lot of people are looking at this issue, a lot of people are thinking about it, and there's a lot of candidate genes that have been described that might or might not be a target for therapeutic intervention for this conversion that ends up being the lethal event for most prostate cancer patients. What did we do? Well, prior to this time, we had been working on a set of techniques to use specific genetic disruptions introduced by retroviruses or lentiviruses into the prostate epithelial cells of the mouse. And after some years of doing that, gaining knowledge of how to handle the cells and develop the system, we applied it to transforming human epithelium from the normal cell tissue areas of the prostate to generate various models of prostate cancer. There's a bunch of papers that precede this, but this one comes right to the point about this small cell neuroendocrine cancers. Junwook Park and the other authors shown here did the following experiment, and I'll just summarize the data from this paper. A series of lentiviruses, in this case, were constructed that could simultaneously be used to infect populations of cells that expressed high levels of MYC and high levels of BCL2. The BCL2 acting as an anti-apoptotic agent to prevent the cells from dying by acute oncogenic stress as we added all its genetic information. Another vector would express myristylated AKT. That's to keep it bound at the membrane, to keep it highly active. And then this vector has both an inhibitor of the RB protein, short hairpin style, and a dominant negative form of p53. Lots of work in the past, various types of approaches have shown that these very aggressive small cell carcinomas almost inevitably have RB and P53 inactivated in some manner. So we combined all this genetic information and infected it into cells that were isolated from normal areas of the prostate that had this characteristics of basal cells. Basal cells give rise to the luminal cells, which are far more prominent cell type in the prostate, and it was very difficult to get out the luminal cells alive. They tend to die very quickly due to anoikis when you separate their cell-cell contacts. But the basal cells we could get out, infected these lentiviruses, and then we placed them, the euphemistic we here is Jun Wook and everybody else but me, actually, into these organoid cultures, which allowed tissue structure to form, cell-cell contact, and it's sort of a center, a lumen form around with layered cells. And then at about 3 weeks, you could then put these into a xenograft situation, grow up tumors, and evaluate the histology. And the result is striking. It's highly reproducible. You get the same type of thing over and over again with many different independent donors of the normal basal epithelial cells. And what you see up top is the histology of a series of transformants in which we have either the full gene complement I just described or selectively leaving out 1 or 2 genes. And the important takeaway lesson is that if you have all of these genetic influences going into the cells, you get an H&E picture of that small cell carcinoma. You get the markers associated with the neuroendocrine development. And then if you leave out BCL2, the frequency of tumor formation goes down, but you basically get very similar characteristics to the tumor that grows out. But if you leave out one or another of these sort of driver genes, you get less and less neuroendocrine character, more and more adeno character, and less of the neuroendocrine type of markers. And so there's a growth component and also a differentiation component at play here, which leads to the biology of these tumors. We also applied the technique of ATAC-seq to look at the amount of chromatin changes between these alternative outcomes. And the takeaway lesson is that there is a huge number of opening and closing of chromatin sites using this ATAC-seq if you take away the RB and P53 influence. In fact, you need to take away both of them, okay? That's the last lane over there, to show that both of them are having an effect on the ability to differentiate, alter, or rewrite that genome information. And so this system has turned out to be very useful for us in a number of studies, but the most impressive thing is that if you start with a completely different preparation of epithelial basal cells from the lung or the bladder, as shown in this paper, and now other tissues, using the same genetic influences and same strategy, you can drive out the production of small cell carcinoma with neuroendocrine characteristics, which by a variety of molecular approaches as well as the histology are essentially indistinguishable. So I think this may be a common pathway for all epithelial tissues and possibly all epithelial cancers if the patient lives long enough to have this developmental sequence ensue. All right, so this opens up more questions than it answers. And one of the questions was approached by my student, Olga Chen, in her PhD thesis, which is represented by just the next couple of slides, but it was a massive amount of work, in which she asked one of the most important questions in biology is time. How long does it take? Does it take the same time every time? And she was able to do this by setting up this system and taking intermediate time points and then using a variety of analyses, including bulk RNA-seq, ATAC-seq, single-cell RNA-seq. So I don't know how many millions of dollars this slide cost, but a lot. Because I found out after she started that this stuff is really expensive, okay? I mean, nobody tells the lab boss. They just say they want to do it. And then I started getting the bills. So it was a condition of Olga's graduation that she stopped doing single-cell seq. Okay, that was. But the take-home lesson here, in addition to the kind of eyeball estimator, you can go from left to right, early to late in the tumor formation, is that the drive molecularly by RNA expression, starting either with lung or prostate cancer from large databases, is recapitulated in the drift from left to right down to this portion of the curve in the model system. And so we thought, well, maybe we're close enough that we could begin to look at more mechanistic aspects of this. So one of the first things is to think about this as a differentiation sequence, and differentiation to me is largely the accumulation of different transcription factors regulating either the turn off of something else or the turn on of the next set of transcription factors. So this is just a diagram across in sort of a timeline manner of the level of 1,600 transcription factors that were identified and called out in the sequence. And there's some familiar names here. ASCL1, we'll come back to that, some of the NKX genes, et cetera, some of the genes that were on an earlier slide, one-cut SOX, NeuroD1, that also turn up in the study and analysis of small cell lung cancer, for example, which can define subsets of those diseases. So we had this large amount of data, and the question is how to organize it. And I'll just grossly oversimplify what Olga and our main collaborator, Tom Graber, have been able to do with these data sets, and just say that as you go from early to late, you go through major inflammatory characteristic responses, stress responses, then reprogramming, like similar to the Yamanaka effect of reprogramming cells back to a stem-like state, and then finally a neuronal or neuroendocrine-like gene expression pattern. And it all kind of makes sense as to what we started with and what it ended up. But one nice feature came out is that it's not a single pathway only. There's at least two major pathways or projections that one could discern from this set of data. One, this one in purple, characterized by high-level expression of ASCL1. We'll come back to that a couple of times in the talk. Another by ASCL2 or POW2F3, and with another transcription factor that turns up in the subclassification of small cell lung cancer. And so starting from normal tissue through this organoid stage, early tumors, transitional, and then to these endpoints, you get this bifurcation in this model system. There's about 100 different testable hypotheses that pop out of this data involving specific signaling pathways, specific things you might consider for targeting. And I'm not going to try to tell you about all of them or even several of them. I'm just going to tell you about one. And it comes from the thing I actually have enjoyed the most about having an academic lab for a long time is that you don't really think of most of the ideas. You bring in somebody and they have an idea that they want to try out. And so a new postdoc came to the lab, Greg. Greg had training in mitochondrial biology, particularly mitochondrial dynamics. And even though he wanted to learn more about cancer and apply his knowledge to cancer biology, he couldn't stop thinking about mitochondria and respiration and metabolic changes. So he began to investigate whether the transformation system we were using and the outcomes we were getting were interesting, different, unusual in any way that involved the fundamental concept of mitochondrial respiration to produce either high levels of OXPHOS or low. Now, everybody in the field of cancer was influenced by the Warburg effect, and which I note this quote from him. He not only thinks that it was something altered about respiration, but also causes it. Now, I'm not sure that's true, but he still got the Nobel Prize, and I think that's a good deal. So we started with this point of view, and what Greg did is he began to look at the details of this, and a couple of things turned up pretty quickly. When these tumors, Part B tumors, as we call them, named after the oncogene collection that generates them, bifurcate into these two subtypes. They have very distinct bioenergetic profiles. And you can see that from the types of gene expression using large data sets, okay? Or more directly using seahorse measurements to show that what we call type 2, which is the ASCL1 high, was significantly higher by these oxygen consumption measurements than the other type. Took this further and began to say, well, what could be the cause of this? And I had heard about PGC1 alpha many times. It's involved in so many different biological processes. People like Bruce Spiegelman give great talks on it and so on. But I didn't really appreciate that this one gene, this one transcription factor, could work on so many different important cellular pathways, including directly regulating aspects about mitochondrial biogenesis and I think activity. So Greg began to look through the lens of this gene, and he found some really striking differences. This one type of tumor that we generated, this ASCL1 high tumor, was dramatically higher for expression of this transcriptional regulator. And if you looked at the level of this gene ASCL1 correlated to the level of this master transcription factor, it was a very, very strong correlation. High level of ASCL1, high level of PGC1 alpha. All right. We could then, using this biological system we had developed to look at different genetic influences on tumor outcome, evaluate whether or not this pathway regulated collection of things that PGC1 alpha could do. So we could do an experiment in which we knock down or suppress the level of PGC1 alpha, and that's shown along the top. By just adding that into the mix of genes, we would enter into the normal basal cells, and the answer was pretty clear. The expression was knocked down in the organoids, and the tumor take was basically knocked out completely. If you took cell lines that were established from tumors of the different types previously, from the xenografts, grow them in culture, and use them as tools, you could also acutely block that gene and then block the growth to a certain extent of those cells. So this becomes target, pathway, something to understand. If you turned it around and artificially induced early on in the experiment a high level of the PGC1 alpha, again, that's by overexpression of the full-length cDNA for the gene, you increase the tumor take, and interestingly enough, you also increased this mitochondrial content dramatically. That was expected, but you also increased the neuroendocrine differentiation as measured by quite a number of different gene markers here by immunohistochemistry or by the overall RNA-seq analysis with many, many other marker genes. Okay. You could also show that this, not surprisingly, increased the OXPHOS in cell lines that have been engineered to do it. So is this a peculiarity of the system we're looking at? Does it have anything to do with naturally derived cancers, et cetera? I think it actually does. I think it may be one of those big global lessons that's just sitting there in the data that's already available. If you look at different cancer types, and you look at the level of this particular cell, this transcription factor against this other transcription factor, again, the correlation holds, and the different types that show up with this correlation tend to be things like small cell lung cancer, neuroblastoma, and that makes some sense. And then if you look at a depth map, the cell lines which are most dependent, okay, in small cell lung cancer pathway knockouts, highest one is OXPHOS-related genes. And so, again, all of this kind of percolates in that direction, that this may be a generalized way to drive this type of biology leading to these very, very aggressive tumor growth. And so what I hope I've convinced you is that for both prostate cancer, and a lot of work I alluded to but didn't show direct data for, but also lung cancer, bladder cancer, and we now have some evidence in ovarian and a few other places, that normal tissue can be driven to this small cell carcinoma with neuroendocrine characteristic phenotype with an enhanced level of OXPHOS, probably generated through PGC1 alpha. And it's opened up a whole bunch of new questions for us to look at. What to do about it? What to do about this prostate cancer, which is now at least 3 or 4 different kinds of disease. Okay? It's a relatively indolent disease at its initial presentation, even through its first metastatic life cycles, when it still remains sensitive to castration maneuvers, until it becomes way out here at the end, is the small cell carcinoma or mixtures of castration-resistant phenotypes. So I wanted to get back to the second half of that Maloney-Murian sarcoma virus curve, which is, in some cases, cancers can be responded to with the immune system and eliminated based on the principles of immune activation. First thing for immune activation, you need an antigen, and the second thing you need is immune response. I mean, it sounds very sing-song, but those are really the 2 most important things. So first step is for prostate, was there a good, well-expressed uniform antigen? And there is one, and it's been known for a long time. Prostatic acid phosphatase, or PAP, is an excellent target for immunotherapy. Its expression of PAP is in normal tissues, and it persists in the metastatic castration-resistant form of adenocarcinoma. It will turn off or go away when you get this full and neuroendocrine differentiation, but it persists into very ill patients who are no longer responsive to any of the anti-androgen maneuvers. It's a secreted form, and so it's going to be difficult, I think, to use traditional CAR-T methodologies, but presentation of peptide epitopes in the MHC would be possible. And there's already a cancer vaccine called Provenge. It was the first cancer vaccine really allowed in a therapeutic vaccine sense on the market. It doesn't work that well, okay? It's not used extensively. It's still used in some patients. And it's based on the idea that immunizing a patient who already has cancer with this PAP antigen with associated cytokine activation might generate T cells, okay, that could help eliminate the tumor. It was allowed as a registered pharmaceutical based on a very, very large clinical trial which showed a modest life extension among the very large control of the treated versus control group. But PAP has a wonderfully restricted tissue expression, and so it caught our interest for this. The other thing that Provenge vaccine showed, data from it, showed that there were patients whose cells taken after the vaccination could be demonstrated to have PAP-specific T-cell responses using assays like ELISpot. But no one had isolated specific TCRs from those ELISpot type assays or had in any way demonstrated the molecular nature of those TCRs as unique or highly reactive. So we set out to make TCRs against PAP, and the first thing we realized is we needed to define its immunopeptidome. This is a simple sounding thing because you just take the sequence and you put it through the algorithms and you get out an answer, and it's a list of peptides. Well, the problem is that there are many algorithms for doing this, and there are many different answers, and to this day I don't think anybody says that's the absolute best algorithm. So what you do is you apply a bunch of algorithms and you get information that's overlapping and also unique. Second way to do it is to use an actual physical capture of the peptides that are residing in the MHC as a more unambiguous way to say that that's a really likely to be important peptide because we can find it resident within the MHC. And of course, you have to pick and choose your MHC, and we picked HLA-A201 because it's the most prevalent form of HLA in the populations likely to be treated. This paper describes multiple alternative technologies that my student Mao used in collaboration with a whole bunch of people for mass spec and other analyses. And he got out 11 very well-described peptides that can bind and rebind and be used in experiments. And we set out to select cells from non-immunized normal individuals, pooled blood, to look for TCRs that would react with these peptides. And we were successful, and we found TCRs. But at a moment I can only describe as disappointing, and for the graduate student, I would say near suicidal, was that the very best TCR is this one shown in red here in a cytotoxicity assay. Here's the cells growing, the target cells growing just fine, and here's after the addition of our best TCR at a ratio, I can't remember what it was, 4 T cells to 1 target cell. It barely suppressed the growth of the cells. So this was a bummer. I mean, I have to say it was a lot of work to get this far. And I thought, maybe we just need to go back in. But we had already gone through a lot of peripheral blood specimens. We had used multiple alternative ways of looking for these TCRs. And it's probably because this is a self-antigen that the highly reactive clones may have been deleted in the thymus. And what we're looking at were the stragglers that got through that are low affinity for interacting with this particular peptide antigen. But fortunately, a friend of mine, Chris Garcia at Stanford, had been working on unique mutations in TCRs. In fact, I think he did the very first demonstration of this when he was here at Scripps as a fellow, to show that there were hotspots for mutations in TCR at the binding interface of the peptide MHC complex that had unexpectedly large effects on the effectiveness of the TCR. He's gone on to study that. There's a bunch of papers, other people in the field as well, and to refer to these as catch bond interactions. And the way this works, and this is the biologist now trying to describe something that's really structural biology and molecular dynamics, is, and actually you'll like this because it's a fishhook analogy, that when the right mutation is made in that binding interface, that there's an allosteric change, and that literally one bond that was at first an approximation now changes and turns into a very strong, usually electrostatic interaction, and prevents the back reaction from happening. So the dwell time of the cell receptor on the MHC peptide complex is now extended. And in biology, that time is really important in making the effectiveness of the second T cell receptor binding to the cell, okay, and the cell staying bound to the target cell, and the effectiveness of cytotoxic killing. So we went ahead with a nice collaboration with Chris's lab, which is continuing for this and other TCRs. And the results are so pleasing, okay? And in this one slide, I don't know how many people years of work, we took pretty crappy TCRs. I mean, they were down here in this peptide concentration response for an activation marker, and moved them about 3 orders of magnitude to greater and greater sensitivity and reaction. And we could go from things that could barely kill the cells at a effector to target ratio of 10 to 1 to now 1 to 4, 1 to 8, 1 to 12, and kill off every target cell in a reasonable period of time. And even better, they work in vivo. In this particular style experiment, we inject the tumor into a subcutaneous site that has the peptide and MHC engineered into it, and then we inject either the parent TCR-laden T cells or ones with these catch bond enhancements, and it's night and day. Almost no effect on the tumor growth to elimination of the tumor and suppression for quite a long time. So we have a long way to go for this, but I actually think this is a decent clinical candidate because of the properties that have been enhanced and the way it works. And I'm going ahead with multiple colleagues, including John Lee, one of my former fellows who left UCLA, go to the Fred Hutch, but now he's come back to UCLA, who will take this forward into preclinical development and I hope into the clinic for good effect for patients. When would you use this? You'd want to use this, I think, fairly early on in the course of disease. You wouldn't want to use it in lieu of surgery because this antigen is also present on normal prostate tissue, and I think you'd have a significant inflammatory reaction and problem in the prostate. But in those patients post-surgery who are now in that twilight zone, not knowing if their cancer is going to come back, you have a great blood marker, PSA, to find quite sensitively that tumor is somewhere residing in the body and will return to growth. And so in those patients, it would be sort of a minimal residual disease type of strategy. Not too long after surgery, if there's evidence of tumor retention in the body, it would be a good opportunity to use something like this. You wouldn't be able to use cytoablative chemotherapy the way we use CAR T treatment. You'd want to find a way of introducing the cells so that they're behaving more in the mode of immune surveillance, going after a chronically produced antigen. And so there's a lot of clinical development ideas that have to be thought of for this very different application than for a late-stage lymphoma patient being treated with CD19 or CD20 or something else. Okay. All right, so that's where we are on that side of the story. Next. What do you do about those patients that have now progressed to this later stage? They no longer have the characteristics of the original adenocarcinoma. They're growing extremely aggressively, and they're acquiring new developmental sequences of transcription factors and gene expression. Well, about 5 years ago, one of my colleagues, then at UCLA, Yi Zhang, who's a terrific bioinformatics person, started conversations with myself and another colleague, Doug Black, about how to measure splicing in its fullest extent, as well as other RNA diversification mechanisms shown here as a way of finding new targets for immunotherapy. This is field of wonderfully smart people developing new computational tools every other week. And the final take-home lesson has to be, can you define such target antigens, okay? Can you then use different sources of T cells to find a T cell receptor, and that's here to here, and then can you show that with some functional validation that they have the right specificity and right distribution of expression to be useful. This whole project came together with multiple labs. You can see listed here, the Zing lab, my lab, Gay Crooks and Chris Seed at UCLA, who work with a wonderful technique called the artificial thymic organoid. It's a way of growing T cells in vitro and not having negative selection. So presumably we will eventually get T cells out that have greater potency because they haven't been negatively selected in the thymus, as well as a whole series of techniques that we use to fish out these rare T cells we're looking for because the repertoire size is so high, finding that 1 in a million, 1 in even 10 to the 7th cells that we're really interested in becomes a technology enterprise. And we've used a variety of technologies, including one I'll tell you about from Dino DiCarlo's lab that I really like because it's got all the features of common sense and scale that we need for this project. So we got started on this and actually got a funding mechanism from the NIH, the Immune Oncology Translational Network, okay, which was designed for these sort of group activities to support it. I won't try to go through, this is really a summary of the kinds of things that Yizing has done on the computational side, but what I'm impressed by is that it's not like you develop one form of the algorithm and analysis. It's like it has to be constantly improved and improved by two things. One, new postdocs and students writing new script to take care of the thing that didn't work so well in the first version. And the second, the massive amounts of new data that pour into the literature in these large databases that you have to go back and reevaluate because you're looking for rare objects. And of course, the more data there is to look at, more likely we're to find it. And most recently, the biggest change has been the advent of intense long-range sequencing of mRNAs, which makes understanding splicing patterns a lot easier to have a contiguous sequence that defines a specific mRNA and the associated splices within it. So we've used Yizing's continually improving algorithms. They do that work remotely now, away from UCLA. He moved to Children's Hospital of Philadelphia. And then we apply various techniques to look for these rare T cells. He'll give us a prediction, a series of peptides. We get them chemically synthesized, and then we use them. Most ways of looking for the T cells are to look for some combination of activation antigens, okay, and then some particular binding characteristic. You might create a dendritic type cell or an artificial dendritic cell presenting MHC in one or more of the peptides or a pool of peptides. What we found to be really interesting and useful is this technique called nanovials that was developed in the lab of Dino DiCarlo at UCLA. Dino and I are friends. We worked on a number of things together. And he called me up one day and he said, do you think you could just like attend one of my group meetings by Zoom? We want to talk about immunology and whether you think this technology might be. So he describes this technology, which is basically a form of this little, biocompatible material with a cavity in it, and the cavity created by Dino's group to basically hold about one cell, one lymphocyte. And you can decorate the inside of this cavity with anything you want that can be tethered with a biotin, avidin type chemistry. The inside of these is actually gelatin, and the rest is a different biomaterial. And so, of course, for you're interested in T cells, you might have an MHC peptide complex that you're interested in. And there's a lot of interesting chemistries and ways to do this, including something called, developed by Jim Heath's lab, which are artificial trimers where the peptide is actually linked covalently. So you can, at scale, make lots of them, of different peptide and MHC complexes. And you might have here an antibody that could, for example, capture a secreted product, okay? Released from the T cell when that cognate MHC peptide interaction works properly. The cell should bind in there, get activated, secrete something you want to monitor. And what's really nice about this is that the avidity of this is enormous. It's tens to hundreds of thousands of molecules of whatever you want to mix in. So you get a multiplex, high avidity, and of course the affinity can be tailored as well depending on what the binding agent is, to look for these rare T cells in mixtures. Even better, once the cell is bound, it's still in a sterile condition and viable, and you could be incubating it to activate, release these cytokines, which could be measured with secondary antibodies or various detection systems. But the best thing for us is that they could be sorted and then analyzed by everything you can do with sorted cells, including direct sequencing as single cells. And so you can get high capture and high level of analysis. And this turned out to be really important because the things we're looking for are very rare. I'll just show you one nice example. This is a peptide, spliced peptide predicted to be antigenic and then verified by synthesis and not antigenic, but MHC binding, synthesized and then in a specific MHC binding assay verified. Of course, we give it a fictitious name because we think it's going to be important, and we haven't gotten all the patents on it yet. But it's found in epithelial cancer, splice product, and it has a very nice distribution by RNA-seq analysis. These are all cell lines with small cell neuroendocrine characteristics out of a very large database of cells evaluated, and these are normal tissues, okay, for that specific splice event and the antigenic predicted peptide. This is an analysis of RT-PCR for a bunch of cell lines. The peptide was predicted from data, including the cell lines that I generated in that specific prostate model, but then it was found most commonly in small cell lung cancer. So the idea is that you might have an antigen and hopefully eventually a clinical grade TCR that would be used more than prostate cancer patients or even prostate and lung cancer patients. It would be for any epithelial cancer that undergoes this specific splicing change and expresses this peptide in the context of this type of HLA. And so this is about where we are. We've been able to get out some peptide reactive TCRs that generate interferon when placed in the context of the right peptide, not the incorrect peptide, and the system is just churning along. But it was very satisfying to me to see that we could get to this point, and that separate work in Yixing's lab, he's also taken some of the other peptides and generated independent TCRs. So I think as a methodology, right now it's not super efficient, but it does work to give you back the TCR. I think these TCRs will have to be enhanced by using techniques like the catch bond mutagenesis schema before we get to the point we think we've got one that could maybe go into the clinical evaluation. So, in summary, I've tried to show you that this conversion of adenocarcinoma to these very aggressive small cell carcinoma types is important. A lot of the neuroendocrine features can be driven from this perhaps specific role of OXPHOS, and this may apply to multiple cancer types. I think I've convinced you, I hope, that you can get at least selective TCRs reactive to a tissue-resident antigen that may be useful for treating patients at a certain stage of disease. And maybe I haven't convinced you, but I'm getting convinced that the splicing products can provide new and unique targets for treating these very aggressive cancers for which there's really no useful chemotherapy at the present time. And the most important slide, of course, is the fact that all of this was highly collaborative, done with work over multiple years with students and fellows in my group, and then multiple labs at UCLA and other institutions. And I keep thinking about my experience in the consulting in the biotech and pharma industry, and seeing that more and more of what we do in academia looks more and more like what we do in biotech. You've got to have experts in multiple areas to make this crank turn around and have something good come out the other end. So thank you for your attention. I don't know if there's time for questions or you want to move on. It's up to...
We'll do the questions over.
Very good.
Over the drinks outside.
Okay, I'm good at drinking and questioning. Okay.
Hi, everybody. First, I want to say what a great thing that Pete and Scripps does this annual symposium. We're all very excited. I think John was such a huge fan of Scripps, and we miss him very much. But we're also very glad that Dr. Lili Lu has taken his spot on the board of Scripps, and we really appreciate having you. So, this is really cool for me to introduce Dr. Xiaowei Zhuang, and one of the reasons is, among her many amazing accomplishments, I'm excited to do this because I just found out that she was also once introduced by Pierce Brosnan. And so that gives me something in common with James Bond himself. And I'm kind of upset that I didn't come up here with a glass of martini during the introduction. So Xiaowei is an interesting, training. She did her Bachelor of Science in Physics from University of Science and Technology of China, and then got her PhD in 1996 at Berkeley, again in physics, and then did a postdoc with Stephen Chu at Stanford. She's a Howard Hughes Medical Institute investigator and a David Arnold Professor of Science at Harvard University. Xiaowei has pioneered techniques in the field of microscopy. She's the inventor of super-resolution microscope STORM, and in genomics, for example, she developed MERFISH, spatial genomics, and all of this work has had really a profound impact on our understanding of how cells and molecules organize to give rise to complex behavior. Xiaowei has won many prizes, including the Vilsack Prize. She's a MacArthur Fellow, and she has what I think is the best prize in biomedical medicine, which is the Breakthrough Prize. So one thing that struck me looking over her CV, as I just mentioned, is that her initial training was in physics, both as an undergrad and in PhD. And as you know, there's a long history of physicists switching to biomedical research and making huge impact. So from the top of my head, I can think of chronologically or semi-chronologically Erwin Schrödinger, Francis Crick, Max Delbruck, Walter Gilbert, Stephen Chu, her advisor, all physics educated. And now we have Xiaowei to add to that incredible list. And I guess this is a good message to trainees that it's okay to change fields. Often doing that brings you a whole new perspective, what you bring from your own field, and apply it to a new one. So I encourage everyone not to be scared to change what you're studying. I think that's all I have, so I give you Xiaowei. We're so happy you're here.
All right. All right. So, thank you, Artem, for that really, really kind introduction. And I would also like to thank Pete and Artem for inviting me to join this really distinguished and fun event. And I do want to say that I didn't have the fortune to meet John Martin in person, so I don't have fun pictures to show you. So what I'm going to just directly dive into is the topic of this lecture, and that is primarily about a genome-scale imaging method that we developed to enable spatially resolved single-cell genomics. With some technology introduction at the beginning, but primarily focused on its applications to study the molecular and cellular organization of the brain. So, just generally speaking, why are we interested in genome-scale imaging? Or I found that no matter where I move, the lights directly shine into my eyes. So is it okay here? Or do you really want to take a picture of me so that I move away from the spotlight? It's not a good thing? Okay, all right. So, so why are we interested in developing methods to enable genome scale imaging or spatially resolved single-cell genomics? And a simple way of putting it is, as many of us know, that our cells or our body is made of many different genes that are simultaneously in action or acting collectively to give rise to our behaviors and properties and functions and so on. So if you could actually study them simultaneously, you get a holistic picture. But of course, you might find that vague motivation just not satisfying because that's too abstract. So let me actually give you some specific motivations. And we know that our body is made of many different types of cells. And these cells, by and large, share the same genetic code. And yet they have very different behaviors and functions in a large part because different genes are expressed at different levels in different types of cells. So if we could actually do gene expression profiling in individual cells, then what we have in hand is a very quantitative and systematic method to identify and discover cell types in our body. And believe it or not, that although we have been doing biological research for how long? More than a century at least, we still don't know how many different types of cells are there in our body. So this is still important. And that is indeed what technology called single-cell RNA sequencing that many of you are using can do. And we just actually heard application of single-cell RNA sequencing and some complaints about how expensive it is from the previous lecture. So why are we interested in developing an imaging-based approach to do gene expression profiling? And one of the reasons is these different types of cells in our body are not randomly distributed, which is very easy to imagine. You know, just imagine your body. You randomize the position of all your cells. What would it look like? It look like a homogeneous, featureless blob, and then you can't hardly imagine that you will be as smart or pretty or whatever nature that you appreciate as a human being. So that would all be gone. So if we can instead not have to, that's because in single-cell RNA sequencing you do have to, it's by all means a very powerful and transformative technology. So don't get me wrong, I'm not trying to say that it's not great. We actually in the lab use it too. So, but what it does have to do is to dissociate these cells from tissues and then extract RNAs from the cells so that you can put them through a sequencer. And when doing that, this spatial organization that is functionally important get lost. Now imagine that in intact tissue, you do not have to dissociate the cells, but can still do gene expression profiling at the whole genome scale. Then you can identify and discover cell types directly in intact tissues without destroying this functionally important spatial organization. And then you can then directly map out this functionally important spatial organization of different types of cells and how they interact and so on to give rise to function. So that's one motivation, but even at the subcellular level, we know that molecules are not randomly distributed. Even we take RNA as an example, RNAs are not randomly distributed. Their spatial organization is actually an important post-transcriptional gene regulation mechanism. And likewise, if we think about our genetic code bearing material, DNA, as a polymer, this polymer is also not randomly folded. And the three-dimensional folding organization of the genetic DNA is actually an important gene expression regulation mechanism. So if we now have a genome scale imaging approach, then we can get these subcellular spatial organizations of molecules or the spatial organization of different types of cells, in a pretty comprehensive and high throughput or high information content manner. So these are some specific motivations that got us to develop the genome scale imaging approach that enables spatially resolved single-cell genomics. So what is our approach? our approach is actually to multiplex, massively multiplex or highly multiplex measurements of different molecular species in an error-robust manner. And again, in that way of general description, you might also find it abstract. So I will give you a specific example of implementation of approach that we developed called MERFISH to help you or to elaborate or help you understand in the specific context what it means. But before I introduce our approach, let me give you some understanding of why is it challenging to do genome scale imaging, right? So for those of you who practiced imaging, you know that if we're interested in imaging a few genes or a few different kinds of proteins, we use multicolor imaging, okay? You have a red, green, and blue color, and now you can study 3 different kinds of proteins simultaneously, how they interact. Now, if you say, well, I want to do genome scale of thousands of these different kinds of genes together, you say, well, can I do thousands of color imaging? And that is not possible because the spectrum of different fluorophores, they are wide enough to overlap that several different colors is pretty much at most you can do, not several thousands. So we're 3 orders of magnitude away. Now you say, well, maybe alternatively, what can I do is this. What I can do is this. I image one gene, label and image one gene, and strip off or get rid of the fluorophores or extinguish the signal, and then I image a second gene, I extinguish the signal, and thousands times later, I imaged all of them, I stack all the images together, and now I get a genome scale image. I, till this day, don't know whether it's feasible, but I know that if I propose to my graduate students, they'll say, well, shall we get out of here because I want to graduate in time. So because of that, this is really a challenging problem. So then let me try to introduce to you, in this particular specific implementation, how do we do MERFISH to overcome these challenges? So here are 3 words that I will elaborate: error-robust barcoding, combinatorial labeling, and sequential imaging. So in this example, which is using RNA as an example, what we do is we assign these barcodes. Could be different kinds of barcodes, but in this case, it's binary barcodes to genes so that a different gene or different genes gets different barcodes, unique barcodes, okay? And then we use combinatorial labeling let me actually first, we use combinatorial labeling to encode the barcode, to imprint barcodes physically onto the genes, and then we read out these barcodes bit by bit sequentially. In other words, in the first round of labeling and imaging, we only detect those genes, their first bit reads 1 but not 0, and then remove the signal, and then in the second round of labeling and imaging, we detect those genes, their second bit reads 1 but not 0, and like this, it's first bit, second bit, and so on, so forth. And at the end of the imaging course, every cell would look like this. They have lots and lots of dots in it. Each dot corresponds to a single RNA molecule, and each RNA molecule now would have a string of binary barcodes associated with it, depending on which round of imaging I actually detected it. If I detected it, it reads 1. If not, it reads 0. Okay? And then how do I do that within individual rounds by this combinatorial labeling and sequential imaging? And here's one scheme of how to do it. So we find these, we first use a large diversity library of what we call encoding probes that can recognize the RNA molecules of interest. And these encoding probes would have specific sequences that can hybridize with the target RNAs of interest. But they would also have these sticking out sequences that I call readout sequences that are designed based on the barcodes. For example, if I have a 12-bit barcode, I would only have 12 distinct readout sequences in this example. And then the collection of encoding probes that gets delivered to this specific RNA will have a collection of readout probes that are determined by which bit reads 1 for that barcode. Okay, so if this gene has a barcode 1010 blah blah 1, then it would have readout sequence 1, but not readout sequence 2, and readout sequence 3, but not 4, and eventually readout sequence N. So then this collection of readout sequences are then detected by fluorescently labeled complementary readout probes, which are also oligonucleotide that can be binding to readout sequences to detect their presence. So with that in mind, then you can see one really advantage of this approach is the scalability. I was actually often do not even give the answer and ask the audience, but the room is a little bit dark. I can't see people, so I'm going to show you the answer here is if you think about n-bit imaging, how many different genes that you can distinguish, and that is an answer of 2 to the nth. Which means that in n rounds of imaging, you can distinguish, actually, it doesn't even take to n rounds of imaging. If you do 2-color or 3-color imaging per round, it's n divided by 3 rounds of imaging. And then you can already distinguish 2 to the n number of genes. Now, if I say, if I want to image 1,000 genes, rather than having to image 1,000 rounds, I'm going to ask you, how many rounds of imaging do I have to do? 10 for 1-color imaging, 2-color, 5. So you can see that when I propose this to my students, they no longer tell me to get out of here, right? Because all of a sudden, it turned this almost like impossible problem into a practical thing. And actually, they got so excited that now much of my lab actually do research in this area. But I also want to say that there is a unique challenge associated with this kind of multi-bit imaging, and that has to do with error propagation or error accumulation, okay? In every round of imaging, this is a approach called the fluorescence in situ hybridization, and single molecule fluorescence in situ hybridization, actually as pioneered by Rob Singer and Ajahn Raj and so on. This is becoming a very accurate approach and has been used for example, imaging 1 gene or 2, almost benchmarking method because it has been used to benchmark single-cell RNA sequencing results, means that the error is very small, just a few %. But when you take it to the genome scale with this kind of a multi-bit imaging approach, as we designed, 1 bit having 3% error, after 2 bits is 6%. After 16 bits, we'll give you 60,000 different kinds of genes imaged. That's about 50% misidentification rate. Now, if I tell people, hey, wow, I have this whole genome profiling of this brain data, but 50% of the molecules are misidentified, you wouldn't know, like, what are you talking about? So how do we actually then overcome this approach? And we overcome this approach by, the excellent scalability you already get. How do we overcome that? We overcome that by error-robust encoding schemes. And this means these codes are capable of detecting and correcting error. So this is an idea actually borrowed from a very, very distant field that you might know of called digital communications, because you get text messages and whatever these days from your cell phone and so on. These things are transmitted in binary codes, okay? And for those of us who remember the very, very, very early days of receiving some garbled email that actually doesn't mean anything, and they couldn't even make up words together. That's because of error in transmission. A way to overcome that kind of an error problem is by using barcodes that are robust to error, means you can detect and correct the error. So there are different ways to do that. Let me explain to you one way, and that is in the binary barcodes, you do not use all possible binary barcodes to encode all valid messages. In this case, for example, all RNA molecules. You choose a subset of it so that between valid barcodes, multiple bit of error has to or multiple bits has to flip from 1 to 0 or 0 to 1 in order to convert something valid to another thing valid. In this way, you have advantages. One is every bit has a small chance of error, and then having them simultaneously occurring is an even smaller chance. But more importantly, if the number of bits have to flip is large enough, for example, in this case, I call it Hamming distance 4, means it's a Hamming code that 4 bits has to occur. 4-bit error has to occur in order to convert one valid message into another. Then if 1-bit error occurred, you can actually know it's not a useful message. It's an invalid message. You can detect that error. And also, if 1-bit error occurred, you know that it's actually uniquely close to a valid message and at least 3 errors away from all the other valid messages, so that you can actually, with a reasonable chance, correct that to actually get a high accuracy measurement. So with that, then we actually ensured that even towards a very large scale, we can multiplex it in an error-robust manner to get a high accuracy. Was that long-winded explanation that can explain why we named our approach multiplexed error-robust fluorescence in situ hybridization, or MERFISH. And even in our very first implementation of MERFISH, published in 2015, we have already demonstrated genome scale imaging of 1,000 genes. Because I told my students while it took us multiple years, and I always, since the conception of my conception of the idea, and I always feel bad that for these earlier pioneers, their data gets shown in one slide. And at those days, I was very ambitious that if I don't get to 1,000, I cannot convince people it's a genome scale imaging method. So I set as a goal, but I really love these students and postdocs, Kao Chen, Alistair Bediger, Jeff Moffat, and Stephen Wang, to actually stick with me, overcome many challenges, and then delivered this genome scale imaging. And here is a more recent example of a MERFISH image of 10,050 genes to give you an idea of what that kind of image looked like. This is a cell. Individual dots are RNA molecules. They're false colored by their genetic identity. And with this, you can already see this not only beautiful, but you can directly see that the RNAs are not randomly distributed, and then you can appreciate their intracellular distribution at the whole genome scale, which is not a topic of this lecture. And then we also extend that to DNA imaging. As I said, the 3D organization of the genome is an important gene expression regulation mechanism. So we also then extended to multiplex the FISH imaging to DNA imaging. In that case, it has its unique technical challenges, which I won't go into details, but just to give you a good concept, very similar to RNA. Instead of using MERFISH to identify RNA, the genetic identity for RNA and localize it, we use MERFISH to determine the genetic coordinates or genetic identity of a chromatin loci and localize it so that we can trace that into a 3D organization. In this way, this one I also have some, personal, how should I say? I like it in the sense that it not only gives you a 3D genome organization, but at a resolution that we call super resolution, because you might have heard from Artem that prior to I developed MERFISH approach, another imaging method that we developed in my lab that you heard is STORM. Did you mention STORM? I don't talk about STORM in this lecture at all, but STORM does it by imaging a subset of molecules, localize them to overcome what's so-called the diffraction limit. Here, because we image a subset of loci at a time, not all simultaneously, and then we localize them, so eventually it's not only a heavily multiplexed or massively multiplexed imaging to get thousands of genomic loci, they also give us super or subdiffraction limit resolution. That's a side note. So we not only can get 3D genome organization, but actually we can use, and the RNA, we can use this to do multimodal imaging so that we can image the 3D organization of the chromatin and the transcription of RNA both at the genome scale as demonstrated in this work where we image more than 1,000 genomic loci and the nascent transcripts of more than 1,000 genes to measure the transcription activity of these loci, and also multiple protein imaging so that we can study the 3D genome organization and its relationship to transcription, all in the native context of the nucleus, cell nucleus, to understand their relationship. And I will have, if I have time towards the end, to illustrate that. And the last technical bits is we also recently extended MERFISH to actually tackle or image epigenetic properties, which is another very important regulatory mechanism for gene expression, such as histone modifications and DNA modifications. And how we do that here is we use antibody to recognize modifications, such as DNA modifications, and then use protein A fused to a transposase TM5 to bind to the antibody that specifically recognizes the antibody. And transposase can actually cut DNA into fragments, also deliver specific sequences at the end of it, as in, for example, a technology you might have heard of called cut and tag, pioneered by Hennekoff lab, right? So in that case, you have sequencing primers get to the end so that you can do sequencing. And here we put T7 promoters at the end so that DNA fragments can be in vitro transcribed into, or in situ in this case, transcribed into multiple RNA and then amplify the signal, and then we can detect and then anchor that physically, spatially, so that we can detect with MERFISH. And then with that, we can actually look into epigenetic elements such as active promoters, putative active enhancers, and even using the enhancer-promoter spatial organization to infer enhancer-promoter interactions or pairs that might be important for gene regulation. And that's not what I will focus today at all. So the final thing is to say that this is maybe very specific for some people who are interested in applying it to, for example, neurobiology or other areas, that when we started with doing MERFISH imaging, these are thin tissue slices. And in applications, in some, for example, neurobiology, you might want to actually do it to relatively thick slices so that you can actually do neuronal activity imaging, for example, by calcium imaging, and then do MERFISH imaging to find which type of neurons that gets activated, and hence give the functional roles of molecularly defined neuronal types. In that case, you want to do thick tissue imaging of hundreds of microns in thickness. And this is just to show you in a recent work that we can indeed do that, and these are RNA dots, and afterwards you can actually identify their cell type based on their gene expression profile, and then after the identification, we give the cell its false color, and then you can study the cell-cell interactions and so on. So this also gives me a great segue into talking about the applications that we did with MERFISH in this specific area of the molecular and cellular organization of the brain. And we started by studying individual brain regions, and then recently completed a whole mouse brain cell atlas that I will talk about. And then also I will show you that we can not only use that to study model organisms, but also directly looking at human brain. And if time allows, I will talk about 3D genome organization, how they differ in different types of cells in mouse brain. Okay, so I'm going to start with this example, which is always my favorite example, because it is our first application of MERFISH to study cell atlas of a specific brain region. And this was done in collaboration with my colleague, Catherine Dulack's lab. And I also always love to tell you how this collaboration started, and especially if there are students in the audience. And you can see sometimes fun things just happen, even among nerdy scientists. Catherine and I, we've already been working on collaborations using STORM to study, for example, subsynaptic structures and so on. And then one day when I was attending a Max Planck Institute symposium, And then I found that, my colleague Catherine is also in the same symposium. At that time, we haven't even published MERFISH yet. And I thought, I bet Catherine would love to extend our collaboration from STORM to MERFISH. So the night before my talk, I completely changed my talk from a STORM-based talk entirely to a MERFISH-based talk, all about unpublished materials, and then give the talk. And then afterwards, I went to Catherine. I said, what do you think? Do you want to collaborate in this area too? And Catherine said, you bet. So, but she said something afterwards, which she said, Xiaowei, I know what you must have in mind as the first brain region you want to study, and that is a cortical region, because many neurobiologists study the cortex. Let me convince you, we should study hypothalamus. And I said, why hypothalamus? Or she said, for one, you could live without your cortex. You cannot live without your hypothalamus. But, that's sufficient of a reason for me. So that is in part because hypothalamus actually is important for some essential homeostatic functions, such as sleep, eating food, drinking water, thermoregulation, and so on. And also, it's very important for controlling a variety of instinctive or social behaviors, such as parenting, mating, and aggression, that I will get into more. So without going into all the details of that study, let me just show you, because after all, that's relatively early work. I don't want to spend too much time. But this is an early image of MERFISH, where we only imaged 150 genes, a subportion of the brain that we studied, just to give you an illustrative or a visual idea. Even with 150 genes all plotted out this, your eye already cannot see much. So what I'm going to do is only show you 8 genes, and these 8 genes are cell type markers of these major classes of cells. Then you can immediately see these cells look reddish because they are strongly expressing inhibitory neuronal marker GET1, hence these are inhibitory neurons. And these bluish or cyanish cells are excitatory neurons because they strongly express this excitatory neuronal marker VGLU2, and these green ones are oligodendrocytes, and so on and so forth. And of course, with so many genes imaged, you can no longer use eye to really distinguish cell type. You will have to use computational approaches such as graph-based community detection approaches for clustering. And let me actually fast forward just to say that using computational approaches, then we actually identified in this pretty small region nearly 100 transcriptionally distinct cell populations. Even among neurons, about 70 transcriptionally distinct neuronal populations that really form beautiful spatial organization. And this is from anterior to posterior over a distance of only about 0.6 millimeter. You can already see the spatial organization change dramatically. And I do not have time to go into details of this, but just to say that from this, we really had learned really interesting insights of, for example, sex hormone signaling and so on, from inferring from cell-cell interactions. But I want to show you this thing of how, once you have a molecularly distinct populations of cells and their organization, you couldn't help wonder, what are their functional roles? And one of the good things about doing imaging is it's easier to do multimodality investigation to both approach function and approach gene expression, or assess function and gene expression. For example, function by using neuronal activity imaging with calcium imaging or voltage indicator imaging and so on. In this case, we did it even in a very, very simple way that we just used immediate early genes that gets turned on when the neuron gets activated, such as CFOS, to mark which cells are activated by a certain behavior, such as parenting. And then these red cells express CFOS much more strongly than other cells and gets turned on by the parenting behavior. And then from these other hundreds of genes or more than 100 genes that we imaged, we can identify their cell type. So with this kind of approach, it's a pretty high throughput approach to survey a variety of social behaviors, parenting, aggression, mating, and for different physiological conditions such as female, male, virgin mothers and fathers, and so on. And from that, we really learned very interesting things about how genetically encoded cells that comprise circuits that actually control different spatial social behaviors. So I won't have time, of course, to go into explaining all these things, but I want to share with you one anecdotal story we learned so that you can appreciate what kind of information can come out of this kind of a study. And that also started with a very interesting fact that Catherine told me that I never knew before, and till this day still find it shocking. And that is, she said, "Hey, Xiaowei, do you know this thing?" And I said, "What?" He said, if you expose pups to virgin female mice, they already exhibit parenting behavior. And mothers, of course, do that too. But if you expose pups, or if you expose virgin male mice to pups, they do not exhibit parenting behavior. Instead, they attack the pups and can even kill the pups. It's called infanticidal. And only a brief period when they become a father, during that brief window, they can do some parenting behavior. I said, wow, I've never heard of a thing like that. So, very impressive. After years, I still remember that. But in case, don't find that offensive. I'm only referring to mice. Okay, have no intention to extend that towards human, for example. So, but I want to share with you this finding of, in that context, give you some idea of the molecular or cellular basis of this behavior. So you can see that with pup exposure for virgin male or female in this particular hypothalamus region, which I should say called preoptic region, of some of the 70 different transcriptionally distinct neuronal populations we identified, one particular type, an inhibitory neuronal type, which we gave it a very interesting name, insightful name, I14. We have more insights of that because there are interesting molecules that we can identify to be expressed in it that make sense. And that is much more strongly activated than any other neuronal cell type in this region. And then this cell type was also activated when mothers and fathers were exposed to pup. And so we call that parenting cells or parenting neurons. And you can see that that is not activated when a virgin male mouse was exposed to pups. Instead, the other inhibitory neuronal type, I16, was activated, and the same neuronal type was also activated when a virgin male mice was exposed to other adult male that would elicit aggression behavior, and hence this is an aggression-related neuronal cell type. And of course, from the molecular markers of this, we also have some hypotheses of molecules being important for this. So you can see that in a measurement like this, it does give you at least a hypothesis of the neuronal, molecular and cellular basis for certain behavior. So, and of course, I did mention, this insight that Catherine told me that I could live without a cortex, but then after thinking more, I thought I may be able to live without a cortex. I don't want to live without my cortex, so we also went on to study mouse cortex, its cellular organization, with Smurfish, and identify the cell types in it, and then see its beautiful lamina organization. I won't go into details of this study, but I want to mention in this particular work, there's another technical thing that we showed you, and we demonstrated. That is, for neurobiologists, we know that long-range interactions between neurons is very important, right? What are the different types of neurons that are interacted through these long-range synaptic interactions? What we can do is we can actually inject retrograde labels into brain region that will retrogradely trace to the original place, for example, the motor cortex that we study, to see what, to label the cell body of the neurons that project to that region. And then from that, we can actually see that what types of neurons actually project from the motor cortex into other brain regions. I won't go into details of the results, but just to say that with that, we found that the projection of motor cortex neurons to other cortical regions is a multiple to multiple type of network where each target can receive input from multiple different types of neurons in the motor cortex, and then each multi-cortex cell neuronal type can also project to multiple targets. But let me actually then get to this recent work that we actually now mapped the whole mouse brain cell atlas, which I think is a really heroic study of a group of lab members, in particular led by this really talented postdoc, Meng Xiang, that in the lab. And then there we imaged 1,100 genes in 10 million cells. These are all really very large numbers in hundreds of brain slices. And we also selected these genes, let me actually give you some idea. Actually, do I have time? Let me see. Okay, so for those of you who practice MERFISH, if you say, how do you do gene selection? Generally, we do that by both using the knowledge existing in the literature to see what are functionally important genes, but also often with the help of single-cell RNA sequencing to find out genes that are differentially expressed in different types of cell populations. So this 1,100 genes was selected with the help of Allen Institute, my collaborators' team led by Honghui Zhan, who in parallel performed single-cell RNA sequencing of the whole mouse brain and selected these genes. And then we also integrated our MERFISH data with their single-cell RNA sequencing data so that we can directly in situ identify different types of cells based on their gene expression profiles. And this gets a little bit of a technical details of once we do the integration, we can also then impute the whole transcriptome-wide expression of every single MERFISH image cell. I won't go into details of how we do that because of the time constraint. But to say that with this, we observed extraordinary diversity of cells in the whole mouse brain that have more than 5,000 transcriptionally distinct cell clusters or cell populations that belongs to hundreds of different major subclasses or major cell types. And here you just see a few example coronal and sagittal section. In each dot is a cell that is color-coded by their major subclasses. And then here is a movie showing you progressively from anterior to posterior. Here is labeled by subclasses. Here is labeled by that 5,000 cell cluster so that you can see the beautiful spatial organization in the whole mouse brain. And from that, we learned really interesting complex spatial organization. One remarkable thing is that these cells not only are molecularly highly diverse, but also spatially highly diverse in the sense that even at the thousands transcriptionally distinct cell population level, most of these different cell clusters have different spatial distributions in the brain. And we also learned interesting things about spatial modules, gradients along the different areas or different directions of the brain. I won't have time to cover, but I will cover one thing, and that is from this kind of a high-resolution cell atlas, we can, in a very high throughput way, infer cell-cell interactions in a cell-type-specific manner. So I'll walk through a little slowly here. What do I mean? I'm apparently jumping forward before me. So not at the cell-cell interaction level. Let me actually show you some examples of the spatial organization and molecular diversity. Neurons themselves already have extraordinary diversity, more than 5,000 transcriptionally distinct cell clusters belong to hundreds of different subclasses, and both excitatory glutamatergic neuron and inhibitory GABAergic neuron have very, very high diversity and very diverse spatial organization among the different types. And not only that, because we have a variety of neurotransmitter and neurotransmitter-related genes in our panel, and we actually can study the neurotransmitter usage of not only glutamate and GABA, but also these modulatory neurotransmitters such as serotonin, dopamine, and so on, to study their spatial and the cell type distributions. And not only neurons, but also non-neuronal cells are highly diverse, and both molecularly and spatially. Just using astrocyte as an example, of the more than 30 transcriptionally distinct astrocyte populations that we've seen molecularly, each one of them take a highly distinct spatial organization. And now let me get back to this thing. What I said is from this high spatial resolution of where different types of cells are, we can actually infer cell type specific cell-cell interactions computationally. So how do we do that? These are cell-cell interactions that arise from soma contact or cell-body contact or cell-body proximity through paracrine signaling or other short-range interactions if the neuronal process is short, okay? How do we do that? Is we do that by inferring for example, between a pair of cell types, we measure the frequency of pairs of cells between these type of cells that come into proximity or in contact, and then compare to a random distribution by computationally randomize the position, but only locally so that we do not destroy the brain structures such as cortical layers or hypothalamic nuclei and so on. And then with that, then we can actually, in this single work, already inferred interactions between hundreds of pairs of cell types, even at the major subclass level, that include both neuron-neuron interactions, non-neuron-neuron interactions, and non-neuron-non-neuron interactions. And moreover, if we look at the cell types that interact, and then compare the cells that interact, pairs of cells that interact with the pairs of that do not interact, pairs of cells that do not interact, to look at the gene expression difference. In particular, for example, whether ligand receptor pairs, some specific ligand receptor pairs are upregulated in an interacting cell type. We could even infer the ligand receptor basis or the molecular basis for the cell-cell interactions. Or we can also look at the functionally important genes that are upregulated in interacting cells to give some inference of the functional implications of these interactions. And of course, I won't have time to talk about hundreds of pairs of interactions. I'll give you one example that has to do with vascular cell and immune cell interaction. And for example, we observed interactions between endothelial cells and macrophages. And then look at the ligand receptor pairs that are upregulated in these interacting cells. What we see is, for example, one pathway, which is the laminin pathway, is highly upregulated. And that actually makes sense based on literature study because in the base membrane of endothelial cells, laminin actually, that in the base membrane of the endothelial cells has previously been shown or hypothesized to be important for the conversion of monocytes into macrophage. So this interaction might actually be one way of controlling the pool of this important immune cell macrophage in the brain. And then among the genes that are upregulated in endothelial cells that interact with macrophage is cytokine, the specific cytokine. And cytokines are also known to be chemoattractant to macrophages. That might be also a way to actually recruit macrophages into the brain. And similarly, we observed interactions between another vascular cell parasites with macrophages and still find cytokines to be upregulated and lemming pathways to be upregulated. And we also observed interactions between these vascular cells with a different kind of immune cell, in this case the microglia, which is the resident immune cells in the brain. And also interestingly, we found there's different preferences. Microglia seems to be preferentially more interacting with parasites than endothelial cells, while macrophage is the opposite, it more preferential interactor with endothelial cells than was parasites. So just skip a lot of details or other kinds of interactions. Let me just summarize that this is a comprehensive characterization of the different types of cells that exist in the brain, their molecular signatures, their spatial organizations, and their cell-cell interactions. And then importantly, in addition to the insights that we could actually extract in this study with this period that we studied, that we also think this is a valuable resource for the whole neuroscience community. And with that realization, we actually aligned or registered our images to the common coordinate framework of the mouse brain, and then deposited our data in these interactive databases where being interactive, that means you can go there, say, this is my favorite brain region, this is my favorite gene, or this is my favorite cell type. You select that, and then you can find where they are and interrogate that in a direct visual and interactive manner. So I hope that many of you will use these resources. So, and as I said, we can not only do Murphish imaging of mouse brain, but also human brain. So in this study, we actually imaged a very large number of genes, and that is 4,000 gene imaging of a couple of human cortical regions at the middle and superior temporal gyrus. And it started as a purely technology development work, which we want to overcome the specific challenges of human brain imaging, such as autofluorescence, and show how many genes we can image. But then afterwards, it turned into a very interesting biological investigation. And we, in this cortical region, again, found a high diversity of cells, not only for neurons, but also for non-neuronal cells. And of course, being spatially resolved, then we can see the position of every type of cells and see a beautiful laminar organization across the cortical depths. And then here's the thing is, when we finished the study, and then this talented postdoc, Rongxun Fang, said, hey, how about we compare human and mouse and see the difference? And then there we saw some really drastic differences between human and mouse. And I will just summarize a couple of them. Without belaboring you with detailed maps, just to say that across different mouse regions, we found at the major cell type level, the composition similar, but you can clearly see the difference between human and mouse. And this comparison is also, we think, quite fair, because in the mouse region, we now also have the whole mouse cortex can compare. But even in this work, we studied a number of mouse brain regions, which include the auditory cortex, because the human STG contains auditory cortex. Include TA region of the mouse, because the closest analog of human MTG is DEA. And you can see a drastic difference, which is summarized here, and that is the human cortex has 5 times higher glia to neuron ratio, 3 times higher inhibitory to excitatory neuron ratio. Even among the excitatory neurons, the IT neuron, which is the intratelocephalic neuron, which is primarily responsible for intracortical communication, those neuron to non-IT neuron excitatory neuron ratio is much higher in human cortex than in mouse cortex, perhaps showing that the human cortex emphasizes intracortical communications. But also at the cell-cell interaction level, I already explained to you how we infer cell-cell interaction, so I'm not going to go through the methods again. But at the cell-cell interaction level, we see drastic differences. Because it's presented in a different way, let me actually walk you through a little bit of what the map means. Here, each circle means the interaction between a pair of cell types, for example, between microglia cells and layer 2-3 IT neurons. And the color of the circle indicating the fold change between the frequency of interaction we observed compared to random chance, and the size indicating statistical significance. Now, without going into all the details, you can clearly see the drastic difference between human and mouse. And I won't have time to go through all the differences, but we'll give one example of a group of interactions that are different or much more enriched in human as compared to mouse, and that's the interactions between these non-neuronal cells and various neurons. And I will have to first say that it doesn't mean these interactions do not exist in mouse. We do know they do exist in mouse, but it's much more enhanced or enriched in human. And then to walk you through a little bit more, and this is a group of interactions between oligodendrocytes and various type of neurons, much more enriched in human compared to mouse. And you can even visually see that. These red and blue things are inhibitory, excitatory neuronal cell bodies, and green and yellow ones are oligodendrocytes. You can directly visualize often these multi-way interactions between cell bodies of oligodendrocytes and neurons. And these multi-way interactions is much more enriched in human than in mouse. And then an interesting question is what's the implication of that? And we do not really have conclusions of that yet. We have some thoughts, and I'll share with you one thought. And that is, if you think about the neurons firing in our brain, controlling, for example, motions and so on, the energy for firing individual neurons in the human brain, because of the long distance, will have a higher energy demand than a mouse. Mouse is much smaller, right? And these are called paraneuronal oligodendrocytes that can provide energy and metabolic support to neurons. So this is perhaps an evolutionary adaptation for the higher energy demand for firing individual neurons in the human brain. And another group of interactions we observed are these interactions between microglia and various type of neurons, especially excitatory neurons, again, much more enriched than a mouse. And then because we included thousands of genes in the study, many of them are ligand receptor pairs, we could look into the ligand receptor pairs that are enriched in interacting microglia and neurons. And that among these enriched ligand receptor pairs between these interacting cell types of microglia and neurons, a few of them are actually genetically associated with neurodegenerative diseases. So this not only provides potential molecular basis for these neurons, but also potential connections of these interactions to neurodegenerative diseases. Now, final part of this particular work is what we learned is a lot of the differences that we observed between primate, in this case human, and mouse arise from non-neuronal cells, which whether it's the proportion or the strength or enrichment of interaction, which I found particularly interesting. And I may not have time let me just actually do not go into any details of this part, but just to say that because I want to give you some high-level conclusions for we also studied aging to see how aged brain differ from young brain in mouse here, and then found that aging also seems to have induced more pronounced changes in non-neuronal cells than in neurons. And a lot of this has to do with inflammation and immune responses of non-neuronal cells in the brain. But because we have spatial resolving power, we also found that this aging-induced activation of these non-neuronal cells, astrocytes and microglia, were in these couple brain regions we studied, cortex and striatum, these are concentrated in a white manner in a way that's correlated with and perhaps caused by oligodendrocyte inflammation. So, so then let me actually tell you a little bit about this 3D genome organization study of cell type specific 3D genome organization in different types of neurons in the mouse cortex. And this is by combining RNA merphage with DNA merphage, using DNA merphage to map up the 3D genome organization and using RNA merphage to identify cell types. And then we can see how cell types are different between different types of neurons. I knew I might have time problem. So rather than going into these specific things, let me go to the summary slide just to share with you, because I don't want to keep you all here hearing another 10 minutes of stuff and then to go beyond my time slot. But I rather want to just to give you a summary of very interestingly, we found that different types of cells really show a wide spectrum of differences in their 3D genome organization between different types of cells that is strongly correlated with cell type dependent transcriptional regulation or transcriptional activity. This happened at all scales, as large scale as the size of the nucleus of neurons being much bigger than non-neuronal cells that are strongly correlated with a much stronger transcription activity of neurons than non-neuronal cells, to a variety of high-order chromatin structure differences, and the radial organization of active and inactive chromatin that are different between neurons and non-neuronal cells. But I will spend a little time to talk about this thing, also at the high-level summary, to show you that we can also not only look at healthy mouse, but also model organisms of mouse that are model organisms for specific diseases. So in this case, as we know from pioneering work of Huda Zagbi, that MECP2 is a DNA methylation binding protein that are the mutation of this actually causes a neurological disease called Rett syndrome. Okay, so then we actually looked into how MECP2 affect the chromatin organization and transcription activity of cells in the cortex in a cell type specific manner. And before that, let me actually share with you this interesting thing, and that is being a DNA methylation binding protein. And it's natural thought in the early days that this gene MECP2 is a transcriptional repressor. Okay? And then when people knock out this gene, do a genome-wide sequencing study, for example, by Huda and by other people, and then found a surprising thing is when you knock out MECP2, about half of the genes increase their transcription activity, and hence showing it is indeed a transcription repressor. But about half of the genes decrease their transcription activity, showing that it could actually be a transcription activator. And right now, there's still not a full understanding of what's the mechanism behind repression and activation. And I don't claim to say that our study completely solved the problem, but it does shed some light on it. And what we found is very interestingly that MECP2 in neurons will affect gene expression when you knock it out in a nuclear radial position dependent manner. It doesn't affect as much in the non-neuronal cells because it's not expressed as much in non-neuronal cells. So let's pay attention to neurons only. That in the position near the nuclear periphery, then you will find deleting MECP2 actually cause a upregulation of gene expression, means in that part it's a repressor. And near the nuclear center, deleting the MECP2 causes a decrease of the gene expression, means there it's a transcriptional activator. Being such a nice radial position dependent manner makes us feel that there might be a common mechanism that helps with this regulation, not necessarily exclusive mechanism. So what we found, here I'm going to skip a lot of results just to say that we found that MECP2 helps the segregation of active chromatin and inactive chromatin. Okay? And it is known that active chromatin tend to be more in the nuclear center, and inactive chromatin tend to be more in the nuclear periphery. One illustration of it helps the separation of active chromatin and inactive chromatin is if you look around the active chromatin, once you delete MECP2, you found that its active to inactive chromatin ratio decreased. And the opposite is true for inactive chromatin. When you delete MECP2, you found that there are more active chromatin near the inactive chromatin. Means that once you get rid of MECP2, they become more intermixed. And indeed, at the nuclear center, the ratio of active to inactive chromatin decreased. At the nuclear periphery, they increased. So one hypothesis is that after you get rid of MECP2, the originally active environment of gene expression in the nuclear center, because of the mixing, now becomes less active, maybe because of delivery of more repressors there. And at the nuclear periphery, the originally repressive environment now become more active, maybe because of the delivery of more transcription factors and transcription activators or transcription machinery there. And this could provide a common mechanism to explain this divergent effect of MECP2. Again, MECP2 can have other mechanisms of causing active and activation and repressing. I'm not saying that this is the only mechanism. But we found that really interesting that there's this common theme that might be able to explain a divergent effect. With that, let me conclude by saying that with spatially resolved single-cell genomics, and then we can actually now get these really high-resolution spatial and molecular organization of organs, such as the brain, and then give you molecularly defined and spatially resolved atlases of cell types, and I showed you both functional and cell-cell interaction annotation. And then because we can query the transcription program and transcription activity or regulation in such a genome-wide, spatially resolved way, and we think it provides a good approach to study the development function of brain and other organs and the dysfunction of them in diseases. And these things, because we can study gene regulation mechanisms such as the epigenome and 3D genome, also at the genome scale and spatially resolved, and hopefully it provides insights into the gene regulation that actually is important for the development function of these organs and how they go around in diseases. So with that, let me actually also get to a very important slide of acknowledging really a wonderful lab of very, very talented and dedicated young researchers, postdocs, and graduate students who has done really remarkable work. And I won't have time to go through all the details and realizing that it's almost an hour. So let me actually skip the people who has really did all the different generations of technology development and only acknowledge those people who I talked about the specific neurobiology applications. And first, as I've already mentioned, that Meng Zhang is really a remarkably talented and dedicated postdoctoral fellow in the lab who led our whole mouse brain study. Actually, I talked about the whole mouse brain study. The most motor cortex study that I talked about was also due to her. And then the whole mouse brain study was in collaboration with Xinjie Pan and Wang Zhang. And this perhaps is not the best time for such a distinct lecture to promote one student, one postdoc. I nonetheless want to do that because I love my people. She's on the job market and for a faculty job. And I asked her, I said, I'm visiting Scripps. Have you applied to Scripps? She said, I applied, and although I received interviews and did interviews at many other places, but not at Scripps. So I wanted to plug in this work. She would be a great catch. So if you still have time to consider her, please do. She's, what I keep telling her is the work, whole mouse brain work she did would be ordinarily, you think about it, would be a paper that would associate with it like 100 authors, that kind of a large kind of a study. And she led that with, just relatively few people. So. All right, and Rongxin Fang is another person that I remarked as a very highly, extraordinarily talented postdocs, and who led the human brain study, also on the job market. I don't think he applied, so. to Scripps, but I'm not sure. Will Allen is another talented postdoc who did the brain aging work, but I think he last year was already accepted a Stanford offer and is going to Stanford. And then the other thing that I did is the 3D imaging, genome imaging. These are led by three very talented lab members, postdoc Shuwei Liu and Cosmos Wan, and a former graduate student, Pu Zhen. And I would like to acknowledge my collaborators, Catherine Dulac, which I have already mentioned for our hypothalamus study, and Honghui Zhen I've also mentioned for our whole brain Sal Ellis study, and at Lane we collaborated on the human brain study, and then Bing Ren, right local here at San Diego, we collaborated on the cell type specific 3D genome organization study, and thank my really appreciative of Hughes and NIH for supporting many years of my research, and the Chan Zuckerberg Initiative for supporting the initial part of our human brain study. And here's my financial disclosure that I'm a co-founder and consultant of Vision that commercialized the Merfish technology. And thank you very much for your attention. All right, again, maybe no questions for the reception period? Okay.
Well, good afternoon, and thank you for your attention. I have the privilege of giving the closing remarks today. I want to thank Pete as well as the donors for supporting this, this memorial symposium in John's honor. I have to say, John was one of my closest friends, so this is very special to me. But it's also special because just in the audience today, I've seen many friends from Gilead with whom I've been affiliated for 3 decades, collaborations, and working with Gilead on several antiviral projects. And also encountering a colleague from virology study section not 1 decade ago, not 2 decades ago, but 3 decades ago, and that's Owen Witte. So it's really a treat to be here. So Scripps has had the luxury in the last 2 weeks of having 3 really spectacular meetings. The first was a symposium that was alluded to by Pete that involved members of industry as well as academia and included Jeannie Marazzo, the new director of NIAID, summarizing some of the interventions that are available for emerging infections, whether it be vaccines or drugs. The second was a review of their AVID grant, all 7 projects, and that was reviewed by a scientific advisory board that was put in place by Arnaud and Sumit. And the third was this afternoon. So I don't want to dwell on the past history. John would never look back. He would only look forward. And he would think more about the future than he did about the past. And I think it's no different today when we think about what John's legacy is. John, as Bill alluded to, was an insightful scientist, a terrific medicinal chemist. He knew how to execute, but he also knew how to drag his friends along with him to get a project across the finish line. And I'm going to use 3 examples today to try and illustrate those characteristics. The first example I'm going to use is a project that involved remdesivir. And when John stepped down as the chairman of the board, he had remdesivir on the shelf and it needed to be developed, but the pandemic had not struck yet. So he and I undertook a different project, and that was to develop a drug for cytomegalovirus infection, recognizing that the drugs that were currently available are either inadequate because of the lack of efficacy, the apparent toxicity, or the propensity to develop resistance quickly when exposed in high-risk immunocompromised patients. That project moved along to the point of doing a patent search, and at the time of John's death, I had to drag Arnaud and the folks at Caliber into the project Now we have to find a way to take really promising lead molecules and push them forward into the clinic. But remdesivir still sat out there, and we need to spend a minute talking about it. I saw Thomas outside, and Thomas in large part is responsible for developing remdesivir to treat SARS-CoV-2 infection. But that was not the first foray into humans, as many of you know. The first foray into humans was in an attempt to treat Ebola, which did not have the same degree of success that it did in the treatment of SARS-CoV-2. So Thomas and his colleagues at Gilead did develop remdesivir. And I would say that if John were alive, remdesivir would have been developed a lot faster, and the orally bioavailable form, obeldesivir, would be available for administration to humans as well. And that would have probably saved the lives of millions of people. But John was interested in more than just the infections that we would typically see, and that is illustrated on this slide. These are emerging infections, and we can't ignore them. We can't ignore H5N1. We're one mutation away from having a highly transmissible human-to-human disease, and yet we don't have adequate therapeutics, we don't have a vaccine to prevent it, and it's sort of like the government sticking its head in the sand and saying, well, it's not going to affect me. Wrong. It will affect all of us. And I predict it's going to be the next epidemic or pandemic the world is about to see. Now, you'll see on this map that SARS does not play a major role in infections that are evolving around the world. And if we simply look at the United States, we have to recognize, guess what's happening? It's not just H5N1, but it's measles that's been imported, and the lack of vaccination in American children on one hand. It's chikungunya in southern provinces of the United States, and that's not a trivial disease at all. And we have to recognize, although we try and ignore it, that dengue is in the United States as well. Now, John would have undertaken the development of antiviral drugs for each of those specific indications as well as many more. But he would also have recognized that there's no way a pharmaceutical company is going to make a significant amount of money developing drugs to treat dengue in the United States. Dengue is a problem worldwide. It's a problem that WHO has to deal with. It's a problem that PAHO has to deal with. H5N1 could well be a pandemic in the United States that we'll all have to deal with. Now, the third example I'd like to use is that of the John C. Martin Foundation. And what's important to remember is John, in his own humanitarian way, wanted to help individuals in the developing world. Traveling around the world, he had the opportunity to meet brilliant, brilliant scientists, but they didn't have the resources to develop their work, whether it's sub-Saharan Africa, whether it was India, or whether it was China. And I'm going to cite three examples that I think represent a new form of science. It's a science that has to prove that there's value in adapting new approaches to whether it's a vaccine or a treatment or antimicrobials. The first was actually prompted by Perwald Olsen, who was on the board with me at Gilead, and he suggested to Lilly developing an antimicrobial resistance project that was being performed in Africa to avoid the problems of bacterial resistance of virtually all infections that were encountered there. That project is underway now. Another project is attempting to develop better drugs to treat malaria, another project that's underway at the present time. A third project is developing an orally bioavailable PD-1 inhibitor in individuals who have small lung cancer, and that too is underway, sponsored by the John Martin Foundation. But one that I'm particularly pleased with is a study that they supported using a simple drug, namely tenofovir, to decrease the vertical transmission of hepatitis B virus infection from pregnant women to their offspring. Why is that important? Well, in Asia, the cost of hepatitis B immune globulin is outrageous. So having a drug that's very inexpensive and can be given to pregnant women can certainly alter outcome under those circumstances. All this is done with a program that was alluded to by Bill earlier, and that's the Access program that John and Clifford Samuel and Greg Alton initiated. Now, John wasn't all work. John did enjoy doing some fun things in life, and I can remember taking him fishing, and this is not a great picture of the rainbow trout he caught, but it was on the Upper Green River in Wyoming. And it was not so much the fish that we caught, but in the evening over a bottle of wine, it was talking about the hatch and whether we read the hatch correctly or incorrectly, and whether or not we really enjoyed being with our friends on the river more than anything else. And I'd leave you with one thought, and that is, remember, John wanted to improve the health of all people of the world. We all need to keep that in mind as we move forward. And if we do that, we'll all be better off. Thank you very much.
Thanks a lot, Rich. And thanks to the speakers. Thanks, Bill, for your terrific comments, always entertaining and insights into John. Thanks, Rich, for a really thoughtful summary of his impact. And to Xiaowei and Owen for really brilliant talks. And I think I think, not only what's great about John is what he did in his lifetime, but I think what's really amazing is you continue to see an impact of somebody like John, even after he's gone. And we look forward to next year's symposia, and we always miss John. Thanks.